Welcome, everyone. So today is November 2nd, 2023. It is our Ask Meeting, Aspiring Scientists Coalition. And I'm very excited to introduce today's speaker, Natasha Cassani. Natasha is a PhD student at the Laboratory of Antiviral Research at the Federal University of Uberlândia. Not sure if I pronounced that right. I hope I did.
Yes.
Natasha works with compounds isolated from the venom of Brazilian snakes and tests them against viruses like Zika virus. This research sounds so incredibly cool. It sounds like it could be like the plot of like a adventure film. It's so exciting, and I feel that whenever there's something like that in research, it's amazing to share it and talk about it. So, Natasha, I appreciate you being here and sharing it with all of us. I will let you take it away. Please feel free to share your screen. Let me know if it doesn't work for you.
Hi, my name is Natasha, and I'm a PhD student at Federal University of Uberlândia. And I'm gonna talk about my research that it's based on toxins isolated from Bothrops jararacussu, venom, and the antiviral findings against Zika virus. Okay, so a little introduction about myself. I am 26 years old. I have a bachelor degree in biomedical science, and my master's was in immunology. And currently, I'm a PhD student at Federal University of Uberlândia in Uberlândia City, under the supervision of Professor Ana Carolina Gomes Jardim. And basically, my research is focused on the drug discovery for emerging diseases such as Zika, chikungunya, and Mayara fever at the Laboratory of Antiviral Research here in Uberlândia, in Minas Gerais state in Brazil. And I'm going to talk a little bit about viruses. I don't know how many of you are into the viruses world, but viruses are intracellular parasites and above 100 nanometers, so they're really, really small. And virus has no cells, but many people talk about if viruses are alive. Are viruses alive? So I'm gonna leave this question for you to answer for me in the end of our talk today. And basically, viruses are made of proteins. So this is the SARS-CoV-2 virus. And we can see in proteins of the structure of SARS-CoV-2, like glycoproteins, such as spike, and the envelope protein that is outside of virus particles. And viruses are have their genome that can be based on DNA or RNA virus and RNA genome. And most of the viruses that we are working now, they are based on RNA genome. And viruses are have a lot of shapes and sizes, such as Ebola, that for me it's the most different virus of all. And Ebola can have like 900 nanometers, but also we can have parvovirus that can have like 20 nanometers. And today we are going to be talking about Zika. So Zika virus is above 50 nanometers, such as dengue and hepatitis C virus. And how to work with virus. So this is me in the lab. We need a biosafety level 2 room, and we can make all the assays and antiviral assays with virus in this room. And we need to use an overall to our own protection and gloves and masks and everything. And let me change here. I'm sorry, this is in Portuguese, but this is what we use in our lab for us to prepare to be able to work with virus. And basically, we need to use masks, gloves, the overall, and we use shoes like boots, and we actually use a second glove, and we look like this, like astronauts in the biosafety level 2 room. And when we are working with antivirals, we can think, where do antivirals come from? How do you discover an antiviral? So we have a very big resource and a very big source for compounds that can have antiviral activities, such as natural compounds. And in this, we have flavonoids that is found in fruits and vegetables mostly. And in this case, we have the quercetin, that is an example, that is the major representative of flavonoids, actually. And the highest levels of quercetin have been found in onions. So we published a paper about the founds of quercetin against Zika, and it showed very good activity against Zika when we tested in vitro. And also we can work with synthetic compounds that is based on a methodology that is called hit to lead. When we have a derivative and we can change the structure of the derivative to be able to see how this compound can work as an antiviral compound when you are changing the structure. So in this work that we published last month, we selected the aminobenzotiazole as a derivative as the initial hit for the optimization against Zika. So benzotiazole have been changing its structure, and we were able to see how those chains can improve the inhibition of Zika when we are working in vitro. And in this case, we have the toxins. And toxins are isolated from snake venoms. And the snake venoms of the venoms of these snakes, they are free of phospholipases A2, toxins, oxidases, and metalloproteases. And we have a snake that it's called Bothrops jararacussu that is found in Brazil, mostly in Brazilian Cerrado. And the venom of this snake are full of phospholipases A2. So in our work, I don't know how to change this. I'm sorry. In our work, we have been working with this phospholipases A2 that it's called Bothrops toxins 1 and 2. So we worked with the two toxins and we were able to see how we could apply the effects of these toxins in Zika infection. So Zika is a flavivirus. I don't know how much people are into virus here, but Zika is a flavivirus and it's very similar to dengue virus. And so this is the structure of the virus. We have the envelope outside of the particle, and we have the membrane protein, the capsid, and the genomic RNA. And Zika was really famous in Brazil in 2015, 2016, that we had an outbreak here in the northeast of the country. And Zika reached 14 states by this time, and almost over 50,000 cases here in our country. And was when the World Health Organization declared Zika as a public health emergency of international concern. And Zika was really the major causative of microcephaly here in Brazil, and we had a lot of cases by this time. And with this, we can work researching antivirals and founding antivirals against Zika. And to work with antivirals, we might start with in vitro assays, and we can use this flask, then we can put cells to grow like viral cells. These are the cells that we use for our antiviral assays, and viral are cells from the kidney of African monkeys. And this is me in the biosafety level 2 room, and I was making an antiviral assay probably. And if you are lucky, you can have those beautiful focus when we are working with great viruses, but this is not the case of Zika. Zika, it's a little bit tricky to work, and we can make the immunofluorescence. Then we have these beautiful images. So this is an image of Zika infection. We can to work with Zika here, we might do the immunofluorescence assay. So basically, when you have a cell that is infected with Zika, you might do the immunofluorescence to tag the protein of Zika with an antibody that is tagged to a fluorescent protein. And we can see those beautiful images where every little cell here that is in green is infected with Zika. And when you have it, you're treated or you're treated control, you can see the decrease of this infection when you do the immunofluorescence assay. And in our work with the toxins, the methodology that we used was with viral cells, and we use a strain of Zika that is from Pernambuco here in Brazil, that is a state from the northeast. And both toxins, Bothrops 1 and 2, were provided by the São Paulo University here in Brazil. And to do those assays, we can make the viability assays, the antiviral assays, and we need to make a dose response assay when we can see which is the effective concentration of these toxins against this virus. And we can make the stage assays to see what is the mode of action of these toxins and which stage of the replicative cycle of the virus the toxins can inhibit the replication. And for all these assays, we might need to do the immunofluorescence, and then you can found the mode of action of these toxins, providing molecular docking or assays with high spectroscopy. So in our results, what we saw at both toxins showed a strong anti-Zika activity. When we do the dose response assay, we can find a selective index that is going to show how much of your compound is strong inhibiting your virus. And we found that Bothrops toxin 1 have an index of 149. And for Bothrops toxin 2, we can see that it had an index of over 10 to the 5. That it's really, really good. When we made the stage assays, we can see which stage of the replicative cycle of the virus compounds are going to inhibit. And we saw that both toxins were able to inhibit Zika entry into the cells. So basically, we add the toxins and the viruses into the cells for 2 hours, and then we take it off to see if the toxins are going to block the entry of the virus into the cells. And we saw that both toxins were able to inhibit this entry almost 100%. And when we look to the virucidal assay, that basically we need to incubate the toxins with the virus for 2 hours, and then we can add it to the cells to see if the toxins are going to be able to connect with Zika particle and then inhibit the infection. And we saw the same when both toxins were able to inhibit almost 100% of Zika infection. And we also can see if those toxins can protect the cells from Zika infection. And what we saw was the Bothrops toxin 2 can because when you add the toxins before the infection of the virus, you can see the Bothrops toxin too are able to protect the cells from Zika infection, but not Bothrops toxin alone. And it might interfere with Zika replication as well, because when we do the opposite, we add the virus first and then we add the toxins, we can see that both toxins are able to have a post-entry, that is what is called a post-entry effect on Zika replication. And when we perform in silico analysis, it might suggest that both toxins are having interactions with Zika E protein, that it's the interlock protein that is outside of Zika particle. Because when we saw those interactions, we can see that both toxins are connecting with Zika E protein that is outside of the virus. And it might be the mode of action of these toxins, because the in silico analysis suggested that it was a very strong connection. And we realized when we saw those results in the spectroscopy, because both toxins can cause molecular changes into Zika particle. When you look only the results of Zika or only the results of the toxins, when you put them together, we can see molecular changes in wave numbers that can show to us that molecular changes are happening in proteins such as glycoproteins, like the E protein of the envelope of the virus. And to finish, we might think, can the toxins block an antibody detection of Zika E protein? Because if those toxins are connecting to Zika E protein, if you add an antibody that it's the E protein, can the toxins block this detection? And we saw the gas. We made an ELISA assay using Zika particles and an antibody anti-E protein, and we saw that both toxins blocked 100% of Zika E protein detection. So basically, what we saw that both toxins can be connecting to Zika E protein, and when we add an antibody, both toxins can block this connection, so it might be the mode of action of these toxins against this virus. So this paper is published already in the International Journal of Biological Macromolecules. And I might have to thank all of the collaborators, because it was a really hard work, but we made it. And this is our postgrad program here. It's called Immunology and Parasitology in Federal University of Uberlândia. And we are in the Uberlândia city in Minas Gerais state, that is the Minas Gerais state. And this is me in the lab. So this is me doing the immunofluorescence assay with using the microscope, the fluorescence microscope. And this is me and Lara in the BSL2 room, working with viruses that we love so much. And for my acknowledgements, I might thank all of these people in those photos, because they help me a lot. Research cannot be done by ourselves, and we might need a lot of help, a lot of people, and I have to thank you all. And thanks, special thanks to my supervisor, that she's so important to me. And for the ones who might be interested, we have an Instagram account of our laboratory antiviral research that it's called LAPAVI. And we share our lab routine, our papers, our classes. So if you might be interested, please follow us. And a little quote to finish. It's that every brilliant experiment, like every great work of art, starts in an act of imagination. research, most of the parts can be really, really hard, but if you just need to keep going and everything's gonna be all right. So thank you so much. This is my email. I also have an Instagram account and a LinkedIn account. So thank you all and obrigada.
Awesome job, Natasha. Super interesting. You can leave this slide up maybe of your handles and just to have it there.
Okay.
I have many questions for you. I would also encourage anybody in the audience to ask their questions in the chat, or if you want to unmute and be courageous and speak out. But in the meantime, I'll start with a question that I have. I'm curious how it feels for you to be working on and studying something that has the ability to directly improve lives in your country and beyond. Like, how does that impact you as a researcher?
I might say that it's a lot of I don't know the word, but uh, we keep dreaming about to have an antiviral and compound that can really turns into an antiviral in the future when we can make the in vivo assays with animals and do clinical assays and clinical trial assays. And I think that's it. It's really a dream that might one day might be true.
Yeah. I mean, I would imagine that for most researchers, that's the dream, is to do something that can, save lives. So that's amazing. Okay, so now I'll ask you a few specific questions about your research after that very broad question. There's actually two questions in the chat that I'll go to. Ian asked, what risk does this toxin pose to the patient? And of course, I know you're doing these experiments in vitro in cells. But is there any indication? Do you have any data suggesting on how tolerable these molecules might be?
We don't have any data, but toxins can have a lot of effects beside the antiviral activity. And basically what we might need to do is found the activity side of these toxins and turn into a peptide that only have this activity side as an antiviral compound, and it's going to be an antiviral peptide. And this peptide can be synthesized, and then we might need to test it again in vitro. And if the activity still there, we can go on and make them vivo assay.
I see. So what you're saying, it sounds like, correct me if I'm wrong, is that these toxins, they show you that something about them is antiviral, and if you can just figure out which pieces are antiviral and keep those and get rid of the toxic parts, that would be future. Yeah, cool. And Ian had another question. Do you know if these toxins could be used against any other viruses? Have you tested them against anything?
Yes, they have a very strong antiviral activity, and we don't have the work done yet, but we saw that they are able to inhibit alpha viruses as well, like chikungunya and malaria.
By the way, again, if anyone has questions, put them in the chat. I have, like I said, several, so I can keep this conversation going for a while. But okay, I thought it was just flat out incredible that you showed that under normal conditions, just in PBS, phosphate buffered saline, just a normal fluid, the virus infects like 100% of cells. But when treated with these toxins, it went to essentially zero. I mean, as a researcher, to see results like that, where you're taking something from 100 to 0% is unbelievable. I mean, usually we're lucky if you see something like, 20% to an 18% change, it's like 2%, hey, we'll take it. So seeing that big of a difference is amazing. And then you showed that, forgive me, I'm going to butcher this, but this specific domain of the virus that the toxins are interacting with is E something, E protein or something like that, right?
Protein, yes.
Protein. So I'm curious. So then, and again, forgive me if you said this, but is it then believed that the toxins are killing the virus? I know you proposed the question, are they alive?
Yeah.
Or is it simply disabling them from entering cells?
Yeah, probably disabling because toxins can have a really strong activity in membranes, and we are not able to see it, but we really think that it's disabling the membrane of the virus, and the virus cannot infect the cells.
Okay, interesting. And
Yeah.
it brings up another question then. I'm curious, like you said again, is a virus alive? I'm wondering then, I understand the generally not very well, but the idea of a virus that, they carry a sequence, that they're bringing into the cell and then using your cell's machinery to amplify that sequence. So I want to propose a poll to the group, and I'm going to do it right now. Are viruses alive? And I want to see what people think.
Yes, I love this question.
Okay. Whoops. I don't know why it says that. Okay, there's a poll available. I'm not sure if it's
Oh, we really need to answer.
Yeah, you have to answer. I want to know what people think.
Okay.
For some reason, I can't answer. I'm pretty surprised by this result right now. As people's answers are coming in, I'm curious, Natasha, what your answer is.
I'm probably gonna say no, but there is no right answer because I'm gonna wait till people answer and then we can talk about.
Yeah. It's similar to like a question of consciousness, being a neuroscientist, that's a question that comes up all the time. Like plants, let's say, can learn. They can learn when the environment around them changes and they can adapt and stuff. So are they conscious? Is that a form of consciousness? These mysterious questions that.
And are they conscious?
I don't know. I mean, it depends on how you define consciousness. To me, I define personally consciousness as the ability to interact with your environment and learn from it and alter your behavior. So by my definition, yes, plants are conscious. But also that would mean that like ChatGPT is conscious, which is not true based on other definitions of consciousness. So anyways, okay, now that we've given people some time to vote, now I'd love to know your answer, Natasha.
Okay. There's no right answer, but some researchers say that viruses are alive because they have their genome and they are able to infect and infect again and everything. But some researchers say they know because they have no cells. So to be alive, you might have cells. So this is my answer.
Yeah, it's interesting. And I gotta say, your presentation, that intro slide was the first time I ever thought about the fact that viruses don't have cells. They're just a capsid with a DNA sequence inside, right?
Yeah.
Yeah, so that's super interesting. So if let's say, just entertain me for a minute, if we classify viruses as being alive in that their life function is to infiltrate cells and produce cause a bunch of their DNA to be replicated, what do viruses then die when they can't enter cells? Like what happens to a virus that's trapped outside of the cell because, let's say, an antiviral is there?
Uh, you say about when the viruses are outside of the cell?
Yeah, so like in the case of your research, your toxin, like you said, you think that it disables them from entering cells. So then
Yes.
what happens to the virus? Does it stay there? Does it die? Like does it
Yeah, if you think the toxins are disabling the membrane of the virus and the E protein, uh, the might be the particle are disintegrating, and this is not gonna be able to infect the cell.
Mhm.
But when you think about the whole particle of the virus and when it's infecting the cell, you need to know that the virus is a intercellular parasite, so it needs the cell to replicate.
Interesting?
Um, we have the
So
answers.
yeah, and there's the poll. So most people agree with you. I
Yeah.
will say I didn't have a chance to vote, but I would have voted yes. And I'm not a virologist. Um, okay, let's see. How do I get this off my screen? Okay, so all right, I have a big question that I've been just dying to know. You may have answered this.
Mm.
How do you isolate these compounds from snakes?
Oh, that's not us. We have a collaborator in University of São Paulo,
That's right.
and they work with the snakes, and probably I'm gonna try to say, but probably they take off the venom, and it might be to turn into powder. I don't know the word. Turn into powder, and then they can isolate the proteins there. It's in the venom,
Hmm.
but I don't have the right methodology for this.
No, that's okay. Yeah, you oh, freeze dry, Ian says. Um,
Oh,
yeah,
really?
you forgot. You did mention that at the beginning that your collaborator sends. Um, Anna says viruses challenge our concept of what is
Yeah,
good. Yeah,
that's true.
super interesting. Now I asked on Instagram what questions people have, and someone
Mhm.
asked, what was the biggest challenge working with Zika virus?
I might say that it's Zika because Zika it's hard to work with Zika when you need to do a lot of assays and you don't have enough virus because Zika has a really slow replicative cycle. So you need to wait like my methodology today I use 70 hours. So it's gonna a lot of time to wait until your result and then you need to do it again. So I'm
Yeah.
afraid it's the virus.
What you
But
would, sorry, go on.
no, but I was saying that I love working with Zika.
It's a funny sentence to the non-scientist. Love working with Zika. Yeah, I mean, you would think that the fact that it takes so long, wouldn't that make it a bad virus? Like, wouldn't that make it bad at infecting human beings and animals?
uh, yeah, when you have a virus that the replicative cycle is really fast, and you might think that in humans it might cause uh, worse disease, but in humans everything is so different. So
Yeah. Um, do you get like used to handling such dangerous samples? Or because I'm assuming when you first joined the lab and you did your first couple of experiments, you were probably terrified and really,
yeah,
really careful. And after a few years of that, does it become kind of just natural? You come in, you put on your equipment, you just like whatever, it's Zika virus.
yeah, no, it's we get used to
Yeah, yeah.
yeah,
That's the funny thing I've found in my own research as well is like things that strike you as like shocking at first eventually just become like a part of your work.
yeah, yeah, it's a very routine.
Yeah. So here's the last question I have for you, and if any others come in through the chat, you
Mhm.
can answer those. Anyone, please feel free to throw them in. Um, what do you think is the biggest sort of remaining question in this field of research, or any big questions that you're curious about related to any of the components of your research. It could be related to Zika virus, it could be related to these toxins, it could be related to how they work. What's sort of on your mind and keeps you up at night?
Uh, Probably if the toxins is gonna be able to inhibit Zika in humans.
Yeah.
I think they are they're gonna be able, but we might wait for this answer.
Yeah. Well, with such compelling results, I mean, 100% to 0%, like even if 10% of that power is retained in human beings and it goes from 100% to 90% in vivo, that's still great. Like that's still really strong. I
Yeah,
would,
yeah.
so would it be accurate that the biggest barrier might be like toxicity, that like getting them to retain their antiviral properties without being toxic?
Yes, absolutely.
Yeah. And how many years do you think it might be before those human trials come together?
Now, we might need to try in animals first, but if you put on paper the whole process to become an antiviral, I might say like 10 years. It's a
Wow.
really, really long time.
Yeah. Well, it's important research like yours that keeps this whole process of science and medicine churning forward. And 10 years might be a long time, but um, to look back 20 years from now and say, wow, hey, we had 10 years of
Yeah,
treatment with this compound versus, you know, 20 years of no treatment, it's always going to be better in the long run. So
yeah,
I hope to see it all move in that direction.
me too.
Well, that's all the questions that I have for you. I mean, I'm very grateful for your time and for you sharing
Yay.
your research with us. Did you have anything else you wanted to share or talk about?
No, I would like to thank you for taking me. I would like to thank you, Julia, that it's here. She's the one who introduced me to the aspiring science. So thank you, Julia. And everyone that is here watching, thank you so much.
Yeah, thank you. And thank you, Julia, as well, for making the introduction. You've been a long time member and I appreciate you. So thank you very much. Natasha, of course, thank you so much again for joining us and sharing this
Thank
exciting
you.
research. I'll be tracking it.
Okay. Thank you.
All right. All right. Take care, everyone.
Bye bye.
Bye.